ChemBot is a robot arm with 6 joints and a 480-degree rotation range. It can perform complex tasks like sample preparation, cell culture, and analysis independently. It supports real-time remote control for safe execution of dangerous experiments and uses equipment like burettes and pipettes to deliver similar results without human intervention. In this video, we will demonstrate an experiment using ChemBot to synthesize gold nanoparticles and observe the changes in their size and the color of the solution depending on the amount of sodium citrate. ChemBot is located in the analysis room of R311. The configuration of ChemBot system is as follows. At the center of the workspace is the robot arm. In front of the arm are a hot plate, a holder for vials, and pipette tips for the experiment. The right side of the arm is a micropipette and a tip collector, while to the left is a controller and an end effector exchange device. Care should be taken not to access this workspace while the ChemBot is operating. In case of a malfunction, you can press the emergency switch to stop ChemBot's operation. The following are the materials needed for the gold nanoparticle synthesis. 3 vials containing 10 milliliters of distilled water and a stirring bar, an aqueous solution of 50 millimolar chloroauric acid to provide gold ions, and an aqueous solution of 1% sodium citrate as a reducing and capping agent. First, turn on the monitor by pressing the power button on the back of the monitor. Next, launch Visual Studio Code from the desktop of the main PC and press F5 to activate user mode. Turn the red button on the controller unit clockwise to have it pops up, then press the green button to make ChemBot operational. Click Run Step 1 in the user window and ChemBot will turn on the hot plate with the 3 vials placed on it. Wait until the distilled water in the vials reaches the appropriate temperature. After approximately 15 minutes, when the distilled water in the glass vials reaches the appropriate temperature, click Run Step 2. The ChemBot will attach the 200 microliter micropipette to its gripper. Click Attach located at the top of the step window, and ChemBot will attach a pipette tip to the micropipette. Click Run Step 3, and ChemBot will transfer 50 microliter of the 50 millimolar chloroauric acid solution into each vial, then remove the used pipette tip. Change the number in the Attach field to 2, then click the Attach button. ChemBot will attach a pipette tip from the next slot. Click Run Step 4, and ChemBot will transfer 50, 100, and 200 microliter of the 1% sodium citrate solution into each vial. Click Run Step 5, and ChemBot will remove the pipette tip, detach the pipette onto the holder, and return to the standard home position. Press the red button on the controller unit to stop ChemBot's operation. Then, click the Quit button in the user window and close Visual Studio Code. Observe the color changes in the solution over the next 30 minutes to monitor the progress of the reaction. Here are the results of measuring the absorbance and particle size of the 3 gold nanoparticle samples using a UV visible spectrophotometer and dynamic light scattering. The UV visible spectrum on the left shows that the maximum absorption wavelength shortens as the amount of sodium citrate increases. The DLS measurement results on the right indicate that the particle size of gold nanoparticles decreases with increasing amounts of sodium citrate. This aligns with the theory that sodium citrate stabilizes the colloidal gold nanoparticles, leading to smaller particle sizes. As demonstrated in this experiment, ChemBot can conduct experiments using laboratory equipment without human intervention, producing results comparable to those obtained by researchers.